The overall goal of this procedure is to acquire long-term observations of a plant root system using light sheet microscopy. To achieve this, it is essential to have a gentle sample preparation method, controlled growing conditions, and an imaging technique that does not alter plant development, such as light sheet microscopy. To begin, plants are grown on the surface of a 2 mm thin layer of gel. The gel with an Arabidopsis seedling is then cut out, scooped up, and transferred onto the microscope sample holder. Inside the microscope, the plant grows in its natural upright position. The roots grow in a liquid medium, while the leaves remain in the air. A lighting system illuminates only the leaves. The water surface is covered in order to keep the roots in darkness.
One of the main advantages of this technique over existing methods, such as embedding the root inside of a gel, is that we are used to grow plants on the surface of a gel. Therefore, we can stick to the very same growing protocol as before. And second, the root is in direct contact with the liquid medium, which can be exchanged for drug treatments, for example.
Pour 30 milliliters of freshly autoclaved half-strength MS medium containing 1.5% phytogel in a square petri dish to create a layer of about 2 millimeters. Sow sterile seeds side by side with at least 1 centimeter space in between. Then, wrap the plate with a micropore tape. Cultivate the plate in your growth incubator vertically for up to 10 days. In this experiment, we use 6-day-old plants.